Hello, my name is Michael Petrus and I'm an Associate Professor of Biochemistry at the University of Missouri. My research focuses on the essential micronutrient copper and how errors in copper absorption and distribution in the body can cause disease. Copper is an essential trace element that is required for human health. It is solely acquired through the diet. Although we need copper at all stages of life, it is particularly important for very young children. The liver copper concentration of a newborn baby is 4 times that of an adult. This high liver copper concentration is thought to supplement copper intake from breast milk, which can be quite variable. However, certain genetic mutations can prevent infants from acquiring adequate copper, and the results can be quite devastating. Menkes disease is a pediatric disorder caused by an overall copper deficiency. It typically appears during infancy, the result of a genetic mutation in a copper transporter that starves the affected fetus of copper in utero, and also during the postnatal period. Infants with Menkes disease tend to have weak muscle tone, brittle hair, and nervous system defects such as seizures and mental retardation. Most patients die in the first few years of life. To combat this and related disorders, my lab is working to better understand how copper deficiencies affect different organs in Menkes disease, which will allow better treatments to be devised. Ultimately, we hope to apply this knowledge to the study of copper in other diseases such as cancer and Alzheimer's disease. But first we need to understand how copper is absorbed and distributed in the body. After we eat, the copper that is present in our food is absorbed via the intestine. Here we see the cells, called enterocytes, that line the wall of the small intestine. The little finger-like extensions on top, called microvilli, face the interior of the small intestine. The microvilli are responsible for exposing as much intestinal surface as possible to the nutrients to allow better absorption. If we look at the microvilli close up, we can see, embedded in the cell membrane, a protein known as copper transporter 1, or CTR1. Copper, which has the chemical symbol Cu, crosses the outer membrane of the enterocytes through this channel-like structure of CTR1. By mechanisms that remain unclear, the copper is then passed from CTR1 to ATOX1, one of the few possible intracellular proteins that act as metallochaperones, or copper carriers, to specific target proteins. ATOX1, in this example, will carry copper within the cytoplasm to the Golgi apparatus, shown here in green, which is the location of the ATP7A protein, the protein that is affected in Menkes disease. ATP7A has 2 jobs throughout most cells of the body, and they're both critical. The first job is to transport copper into the trans-golgi network, a major secretory pathway sorting station where certain copper-dependent enzymes are made before being secreted into the blood. The second job is to export copper across the plasma membrane. In the case of the intestinal enterocyte, this export of copper occurs across the basolateral side of the cell. This is the side that faces the bloodstream. Once copper enters the bloodstream, it is available for use by the liver initially, and then ultimately by other tissues of the body. But what happens if ATP7A does not function properly? In the case of Menkes disease, a genetic mutation might prevent the synthesis of ATP7A or create a form of ATP7A that cannot transport copper. Again, we can see that the copper has entered the cell, finds its way to the chaperone ATOX1, but since the ATP7A protein is unable to function, the copper remains outside of the trans-golgi network. The consequence, of course, is that important copper-dependent enzymes are not formed, and copper remains trapped within the enterocytes. This entrapment of copper in the enterocyte starves the surrounding tissues of copper, including the liver and the brain. As a consequence, various copper-dependent enzymes are inactive, resulting in the distinctive symptoms characteristic of Menkes disease. If copper is injected into the bloodstream in order to bypass the intestinal blockage in Menkes patients, there is still a barrier to transporting copper into the Golgi apparatus in cells throughout the body. For years, scientists working to understand the role of copper in cells and to combat Menkes disease have struggled to overcome a very basic problem: how do we study a disease whose sufferers seldom live into adolescence? The best answer involves using an animal research model, but in the case of Menkes disease, mice that carry the same types of mutation in the ATP7A gene as those in human Menkes patients either do not survive gestation or die shortly after birth. To move beyond this difficulty, our lab has developed a so-called floxed mouse strain, in which the ATP7A gene has been modified to allow it to be deleted or knocked out in a tissue-specific manner. With this new mouse model, we can determine the importance of the ATP7A gene in specific cell types, and in turn, where tissue-directed gene therapy might one day yield effective Menkes treatments. The University of Missouri has been a wonderful place to carry out this type of work. The general openness to collaborative research among the faculty at MU and the state-of-the-art facilities, particularly in the Life Sciences Center, and of course my research team, have certainly made this type of work possible.